The complement system is a complicated cascade of events, and it's part of the innate immune system. But what exactly does the complement system do, and how does it work? Complement proteins are plasma proteins made by the liver. Before we go into detail of the different pathways, let's first list the main functions of the complement system. One of the functions of the complement system is to produce anaphylotoxins. So these are chemicals which enhance the inflammatory response by causing mast cell degranulation, which leads to the production of pro-inflammatory chemicals such as histamine. It will also produce proteins that will enhance chemotaxis, so it will attract white blood cells such as neutrophils and macrophages to the site of infection. It can also produce proteins which can act as opsonizing agents, which means that the proteins can coat the pathogens and make it easier for white blood cells such as neutrophils and macrophages to phagocytose them. The complement system also leads to the formation of the membrane attack complex, which is a very important structure because it can kill pathogens by penetrating the cell membrane and creating pores, which can lead to cell lysis. So these are the main functions of the complement system. Let's quickly review all three complement pathways and see how they produce proteins which perform all these functions. Remember, the most important step of all three pathways is the production of C3 convertase, as this will lead to the common pathway. So let's see how all three pathways produce C3 convertase. So let's start with the classical pathway and how it leads to the formation of C3 convertase. The classical pathway is triggered by the formation of antigen-antibody complexes. So when IgM antibodies or IgG antibodies bind to antigens, the Fc portion of the antibodies will become exposed, and then C1 will be able to bind to it, and this will activate C1. Activated C1 will cleave C4 into C4a and C4b. C4a is an example of an anaphylotoxin. C1 will also cleave C2 into C2a and C2b. C4b and C2b will come together and form C4b2b. And C4b2b is an example of a C3 convertase. So this is how the classical pathway leads to the formation of a C3 convertase. Let's now discuss the lectin pathway. The lectin pathway is triggered by carbohydrates on the surface of pathogens such as mannose. Mannose binding lectin can bind to mannose and become activated, and the activated mannose binding lectin has a very similar structure to activated C1. And so from this point forward, the lectin pathway will have the same pathway as the classical pathway. The mannose binding lectin will also cleave C4 into C4a and C4b, and remember C4a is an example of an anaphylotoxin. The mannose binding lectin will also cleave C2 into C2a and C2b. C4b and C2b will again come together and form C4b2b, and remember this is an example of a C3 convertase. So this is how the lectin pathway leads to the formation of a C3 convertase. Let's discuss the alternate pathway now. The alternate pathway does not have an initial trigger like the other pathways. It starts with C3b. But where does the C3b come from? It comes from the spontaneous cleavage of C3, and this is why there's always small amounts of C3b present on the surface of pathogens. There are other proteins involved in the alternate pathway. Factor B will be cleaved into factor Ba and Bb. This is done with the protein factor D. C3b and Bb will come together and form C3b-Bb, and C3b-Bb is an example of a C3 convertase. So this is how the alternate pathway forms a C3 convertase. So all three pathways lead to the formation of a C3 convertase. But what does the C3 convertases do? Well, as the name suggests, it will convert C3 into C3a and C3b. C3a is an important anaphylotoxin. C3b is the most important opsonizing agent produced by the complement system. The C3b can also combine with the C3 convertases produced by all three pathways and form C5 convertase. The C5 convertase, as the name suggests, will convert C5 into C5a and C5b. C5a is also a very important anaphylotoxin, and C5a will also enhance chemotaxis. Please note that C3a can also enhance chemotaxis, but its effects are not as potent as C5a. C5b will make a complex with C6, C7, C8, and C9 and form the membrane attack complex, which again was very important in causing cell lysis. So if we finish off this list, the membrane attack complex is formed by complement proteins C5b to C9, and that is the complement system.